Bacteria are prokaryotic cells that play an important role in human disease and health. They can cause disease, but are also part of the human microbiota and live on our skin, body, and on everyday objects in our environment. When compared to eukaryotic cells, the structure of bacteria is less complex due to a lack of nucleus and membrane-bound organelles such as mitochondria, endoplasmic reticulum, and Golgi complexes. Now, bacterial cells are often surrounded by several layers, which are collectively called the cell envelope. Let's start with the cell wall, which is found on the outer surface of the cell membrane, and its most important role is to protect the bacteria. It consists of peptidoglycan, which usually helps differentiate whether bacteria are gram-positive or gram-negative. Gram-positive bacteria have a single thick layer of peptidoglycan above the plasma membrane, which allows them to retain the staining dye. And gram-negative bacteria have a thinner layer of peptidoglycan sandwiched between the surface membrane and the plasma membrane, so they can't retain the dye. Additionally, the cell wall helps maintain their shape. The round-shaped bacteria are called cocci. The rod-shaped ones are called bacilli. Spiral-shaped ones are spirilla. And sometimes the same bacteria can have multiple forms, in which case they're called pleomorphic. Some bacteria are covered by a capsule, which acts as a shield that protects the bacteria against phagocytosis and also helps the bacteria adhere to surfaces. The capsule is considered an important virulence factor since the strains that lack a capsule are less virulent. Underneath the bacterial cell wall, there's the plasma membrane, which is the most important layer because it encloses the cytoplasm, which is a gel-like substance composed mainly of water that also contains cell components, enzymes, and various organic molecules. If the plasma membrane is removed, the cell's contents spill into the environment and the cell no longer exists. Now, the plasma membrane is responsible for most of the cell's relationship with the outside world by acquiring nutrients and eliminating waste, and also maintains the interior of the bacteria in a constant, highly organized state. Usually, all plasma membranes are selectively permeable barriers, which allow certain ions and molecules to pass in and out of the cell while preventing the movement of others. However, in bacteria, the plasma membrane has other important roles, such as respiration, photosynthesis, and the synthesis of lipids and cell wall components. Now, in the bacterial cytoplasm, there are scattered cell components such as ribosomes, which are the sites of protein synthesis. These ribosomes can be found spread all over the cytoplasm, where they produce proteins that are destined to remain inside the cell, or they can be attached to the plasma membrane, and they are called plasma membrane-associated ribosomes. And in this case, they make proteins that will reside in the cell envelope or get transported outside the cell. Another structure found in the cytoplasm is the nucleoid, which is an irregularly shaped region that contains the cell's chromosome. The chromosomes of most bacteria are a single circle of double-stranded DNA, but some bacteria can have a linear chromosome, while others can have more than one chromosome, and these chromosomes contain the genes needed to provide instructions for protein synthesis in the cell. Additionally to the genetic material contained within the nucleoid, many bacteria contain extrachromosomal DNA molecules called plasmids. These plasmids are small, double-stranded DNA molecules that can exist independently of the chromosome, and they can be circular or linear. Plasmids have relatively few genes, and their genetic information is not essential to the bacterium, and cells that lack them usually function normally. However, plasmids often contain genes that confer a selective advantage to the bacterium, such as resistance to antibiotics. Finally, The bacteria can move or attach to other cells and surfaces with the help of structures that can be found on their external surface. So they have pili, which are hair-like structures that allow them to attach to other cells, and when these are shorter, they're called fimbriae and help the bacteria attach to surfaces. They can also have flagella, which are long, whip-like protrusions that allow the bacteria to move. Alright, as a quick recap, the bacteria are prokaryotic cells that play an important role in human disease and health, and can be found almost anywhere in the environment. Bacterial cells are often surrounded by several layers, which are collectively called the cell envelope. They have a cell wall which protects the bacteria, determines whether they are gram-positive or gram-negative bacteria, and also helps maintain their shape. Some bacteria can also have another extra layer on top of the cell wall called a capsule, which is a major virulence factor. Underneath the cell wall, there's the plasma membrane, which is the most important layer because it encloses the cytoplasm and defines the cell, having important roles such as respiration, photosynthesis, and the synthesis of lipids and cell wall constituents. The cytoplasm contains ribosomes, which are the site of protein synthesis, the nucleoid, which contains the genetic material, and plasmids, which contain genes that confer a selective advantage to the bacterium.
With Bacillus anthracis, bacillus means little rod, and anthracis means coal. So Bacillus anthracis is a rod-shaped bacteria that causes a disease called anthrax that's associated with characteristic black skin lesions. Throughout history, Bacillus anthracis, or B. anthracis for short, has caused a number of plagues in Europe, and it's also been used as biological warfare. Not a great reputation. Okay, now B. anthracis has a thick peptidoglycan cell wall, which takes in purple dye when gram-stained. So this is a gram-positive bacteria. Also, it's a non-motile bacteria and a facultative anaerobe, meaning it can survive with or without oxygen. B. anthracis is also a non-beta hemolytic bacteria, because when cultivated on a medium called blood agar, B. anthracis colonies don't cause beta hemolysis, where hemolysis, or breakdown of the red blood cells that surround the colonies, makes the blood agar change color from red to transparent yellow. Finally, Bacillus anthracis is a spore-forming bacteria, so it can undergo endosporulation when it feels threatened by the environment, like when the temperature becomes too high or too low, in case of extreme dryness, or when there's harmful radiation around. Endosporulation means that the bacteria starts by replicating its DNA, and then it forms a wall inside the cell, isolating the big portion of the cell, let's call it the mother cell, from the small portion of the cell. Next, the plasma membrane of the cell surrounds the newly formed small portion, and then pinches it off, forming a separate body known as a forespore. Next, the forespore gets completely engulfed by the mother cell, sort of like a cell within a cell. Finally, inside the dying mother cell, the forespore loses water and accumulates calcium, and at the same time gets wrapped in the super tough cortex from the dying mother cell. At this point, the endospore is able to resist heat due to the presence of dipocalinic acid found in the core of the Bacillus anthracis spore, harsh chemicals, digestive enzymes, and even antibiotics. Finally, as the mother cell dies off, the endospore is released outside. Surprisingly, an endospore can last over 1,000 years out, waiting for favorable conditions to come, and then germinate into the bacterial or vegetative form, which can then grow, divide, and infect organisms, causing anthrax. In humans, anthrax can infect the skin, the lungs, or the gastrointestinal tract. In all cases, B. anthracis enters the body in the endospore form, which gets phagocytosed or eaten up by resident macrophages. For example, when the bacteria reach the alveoli, which are the tiny air-filled sacs where gas exchange occurs in the lungs, They are eaten up by lung macrophages and transported via lymphatic vessels to the nearby mediastinal lymph nodes. A similar process occurs in both the skin and GI tract. Inside the macrophage, the spore germinates, releasing the active form of the bacteria that then exits the cell via cytolysis, or rupture of the cell membrane. Basically, the cell bursts, releasing the bacteria into the surrounding tissues. Now, in terms of pathologies, first, the active form of the bacteria produces a poly-D-glutamic acid capsule that has antiphagocytic properties. So once it's released from the macrophage, it cannot be ingested by another macrophage again. Next, B. anthracis secretes an anthrax toxin, which is composed of 3 proteins: protective antigen, lethal factor, and edema factor. The protein called protective antigen gets secreted from the bacteria and then binds to the surrounding immune cells via a cell surface protein called either ANTXR1 or tumor endothelial marker 8. It turns out the protective antigen gets its name because if a person has antibodies to this protein, they are considered immune to B. anthracis. After binding, protective antigen forms a channel in the immune cell membrane that allows for 2 additional proteins called lethal factor and edema factor to enter the intracellular space. Lethal factor is a zinc metalloprotease, which is a protein that utilizes zinc as a cofactor to cleave mitogen-activated protein kinases 1 and 2. These enzymes help to make NADPH, which is an important cofactor in other biochemical pathways that allow cells to generate energy in the form of ATP. So when lethal factor cleaves these enzymes, cells can't generate ATP anymore and eventually die through apoptosis or programmed cell death. Edema factor gets its name from the edema it creates surrounding the black eschar. It contains ADP ribosyl transferase, which converts adenosine triphosphate to cyclic adenosine monophosphate, or CAMP. CAMP activates the protein kinase A signaling pathway, and thus leads to activation or inactivation of many cellular functions. Importantly, activation of this pathway perturbs the cell's ability to create cytokines, which are single molecules that call for white blood cell reinforcements to neutralize the bacteria. So the bacteria multiply and destroy immune cells in the lymph nodes, preventing an appropriate immune system response, causing surrounding necrosis and eventually bleeding. Access to the bloodstream now allows the bacteria to travel to multiple organs through the bloodstream, causing even more necrosis, leading to multi-organ failure, septic shock, and death. Clinically, anthrax has 3 forms. Skin, or cutaneous anthrax, is the most common form of the disease and results when endospores come in contact with an open break in human skin, usually via animal hair or wool. Here, the endospores also germinate locally, producing both additional spores as well as active bacteria that form painless small red bumps or papules that eventually become a large vesicle, or a fluid-filled sac that ruptures. After rupture, the surrounding tissue begins to necrose or die, leaving behind a characteristic painless black skin lesion called an eschar. Gastrointestinal anthrax occurs when a person ingests undercooked animal meat infected with spores. The ingested spores can affect any part of the GI tract from the mouth to the rectum, and through a similar process to that of the skin, they can cause the formation of black eschars. Symptoms include abdominal pain, nausea, and vomiting, especially vomiting blood, and diarrhea or bloody diarrhea. Respiratory anthrax, also known as wool sorter's disease, occurs when a person inhales anthrax spores. However, in this form of disease, black eschars do not appear in the lung, but instead in the surrounding mediastinal lymph nodes. This can lead to pulmonary hemorrhage, severe mediastinal lymph node necrosis, and bacteremia, which is when the bacteria enters the bloodstream. This results in coughing up bright red blood, high fevers, low blood pressure, and a fast heart rate. So unfortunately, this form of anthrax is associated with the highest mortality rate. Diagnosing anthrax can be difficult because of its rarity and the fact that most hospitals are not equipped with the proper diagnostic tools. The tools that are used to diagnose anthrax include a polymerase chain reaction assay that directly detects B. anthracis DNA, immunohistochemical staining of tissues, which identifies B. anthracis antigens in tissues, and an antiprotective antigen immunoglobulin assay, which identifies antibodies against protective antigen. Other clues to diagnosis include a widened mediastinum on chest x-ray due to progressive mediastinal lymph node necrosis. Treatment of anthrax involves intravenous antimicrobial combination therapy, anthrax antitoxin, and supportive care. First-line antimicrobial agents include ciprofloxacin, along with either clindamycin or linezolid. Finally, raxibacumab and obiltoxaximab are two monoclonal antibodies that directly inhibit the protective antigen and should be administered to anyone confirmed of having fulminant systemic anthrax. Alright, as a quick recap, Bacillus anthracis is a rod-shaped, gram-positive, spore-forming organism that causes a disease called anthrax. The spore form of B. anthracis allows the organism to survive extreme environmental conditions, and it's also the infectious form of the bacterium. B. anthracis causes respiratory, gastrointestinal, or cutaneous disease, using virulence factors like protective antigen, lethal factor, and edema factor. All three forms can progress to fulminant disseminated anthrax. Confirming the diagnosis is done with a positive polymerase chain reaction, immunohistochemical staining, or antiprotective antigen immunoglobulin assay. Treatment of anthrax involves intravenous antimicrobial combination therapy, anthrax antitoxin, and supportive care.
With Bacillus cereus, or just B. cereus, Bacillus means little rod, and cereus means wax. So Bacillus cereus refers to a rod-shaped bacteria that looks like a wax candle. Most often, this bacteria causes food poisoning, but it can also cause more serious infections, mainly in immunocompromised individuals. B. cereus has a thick peptidoglycan cell wall, which takes in purple dye when gram-stained, so this is a gram-positive bacteria. Also, it's a motile bacteria, since it has thread-like flagella that can help it swim in a liquid media, or swarm on a solid surface like culture media. It's also aerobic, meaning it requires oxygen for growth. B. cereus is catalase positive, meaning it produces catalase enzyme. This can be tested by adding a few drops of hydrogen peroxide to a colony of bacteria, and catalase makes hydrogen peroxide dissociate into water and oxygen, making the mixture foam. It's also a beta hemolytic bacteria, because when cultivated on a medium called blood agar, B. cereus colonies cause beta hemolysis, also called complete hemolysis. That's because B. cereus makes a toxin called beta hemolysin that causes complete lysis of the hemoglobin in the red blood cells, making the blood agar change color from red to transparent yellow around the colonies. Finally, Bacillus cereus is a spore-forming bacteria, so it can undergo endosporulation when it feels threatened by the environment, like when the temperature becomes too high or too low, in the case of extreme dryness, or when there's harmful radiation around. Endosporulation means that the bacteria starts by replicating its DNA, and then it forms a wall inside the cell, isolating a portion of the rest of the cell. Let's call it the mother cell. Next, the plasma membrane of the cell surrounds the mother cell and then pinches it off, forming a separate body known as a forespore. The forespore then invaginates into the mother cell and gets completely engulfed by it. Inside the dying mother cell, the forespore loses water and accumulates calcium, and at the same time gets wrapped in a super tough cortex from the dying mother cell. At this point, the endospore is able to resist heat, harsh chemicals, digestive enzymes, and even antibiotics. Finally, as the mother cell dies off, the endospore is released outside. Surprisingly, an endospore can last over 1,000 years out, waiting for favorable conditions to come, and then germinate into the bacterial or vegetative form, which can then grow and divide. So these endospores can also contaminate food like rice or pasta, and some of them germinate into vegetative bacterial cells, which then start producing a toxin called cerulide within food. Cerulide is a toxin known to cause vomiting when ingested, so it causes an emetic type of food intoxication. Cerulide is also heat resistant, so even when food is cooked or reheated, the toxin is there to stay. If someone eats that cerulide contaminated food and gets them into their gut, Cerulide binds to gastrointestinal mucosal chemoreceptors, and then excessively stimulates the vagus nerve, thus activating an exaggerated vomiting reflex. Aside from cerulide production, the endospores themselves reach the small intestines, where they germinate into the vegetative form. Here they produce other toxins, called enterotoxins, because they affect the intestinal epithelium. There are 3 types of enterotoxins: hemolysin BL, non-hemolytic enterotoxin, and cytotoxin K. All of these enterotoxins destroy the intestinal epithelium, triggering secretion of electrolytes, like sodium and potassium ions, and water into the intestinal lumen, resulting in watery diarrhea. Finally, B. cereus can also contaminate medical devices, like needles and prosthetic devices like central lines and indwelling catheters. From such devices, bacteria can get into the bloodstream, causing bacteremia. From there, they can spread to other organs and cause infection, mostly in immunocompromised individuals, like those with an underlying malignancy, or in newborns who have an immature immune system. If they spread to the heart, they cause endocarditis. If they get to the bones, they cause osteomyelitis. And if they make it all the way to the central nervous system, they might cause meningoencephalitis. Finally, if B. cereus can also spread to the eyes, causing endophthalmitis when they get to the interior of the eye, or keratitis when they invade the cornea. Symptoms of B. cereus food intoxication depend on the type of infection. With the emetic type, there's nausea, vomiting, and abdominal cramps. And all of these classically start 1 to 5 hours after someone has eaten cerulide-contaminated food. With the diarrhea type, there are moderate to severe abdominal cramps, and it typically starts within 6 to 18 hours after ingestion of the endospores. With bacteremia, there are signs of systemic infection, like fever, chills, and malaise. Additionally, with endocarditis, there might be night sweat and new cardiac murmurs. With osteomyelitis, there's bone pain and swelling. And with meningoencephalitis, there's a headache, confusion, and stiff neck. Finally, with endophthalmitis, there can be a severe eye pain and swelling, and the eye can even bulge out of its socket, which is called proptosis. With keratitis, there might be impaired vision. Diagnosis of B. cereus food poisoning is done by isolating bacteria from the vomitus or stool. The presence of B. cereus is confirmed only if there are more than 10 to the 5th power colony-forming units per gram of biological sample, or if more than 10 to the 5th power colony-forming units per gram are isolated from possibly contaminated food like rice or pasta. Another way for testing for B. cereus is to look for its toxins in the food, using assays like latex agglutination tests, immunochromatography, and testing for the presence of the bacteria's genetic material using polymerase chain reaction, or PCR. When a systemic infection is suspected, blood cultures are done to identify the bacteria in the blood. Treatment of B. cereus infections usually consists of only supportive treatment for foodborne illnesses. With invasive infections like bacteremia, treatment is done by first checking if there's an indwelling medical device suspected to be the source of the infection, and then that device is removed or replaced. Next, antibiotic therapy is initiated, and vancomycin is the drug of choice. But imipenem and gentamicin can be alternatively used. Alright, as a quick recap. Bacillus cereus is a rod-shaped, gram-positive, beta-hemolytic, catalase-positive, spore-forming aerobic bacteria. It produces a toxin called cerulide, which causes the emetic form of food intoxication, and 3 enterotoxins, called hemolysin BL, non-hemolytic enterotoxin, and cytotoxin K, which causes the diarrhea form of food intoxication. Sometimes this bacteria can contaminate medical devices and cause bacteremia and a number of infections to immunocompromised people, like endocarditis, osteomyelitis, meningoencephalitis, and eye infections. Diagnosis of B. cereus infections relies on identifying the bacteria in biological samples, or using assays to test for the presence of its toxins. Treating B. cereus infections mainly consists of supportive care in cases of food intoxication, but for systemic infections, antibiotics like vancomycin are used.